In preparing for lab final exams, it can be helpful to go back through the 2 projects we did this semester, the Yeast UV and the Choosy Worm project. And for each, I suggest that you think about what was the big purpose, what question were you asking, and what experimental approach did you use to answer that question. Consider your independent-dependent variables, any controls, and the techniques that you used, as well as how you analyzed your data and perhaps presented it. So for your Yeast UV project, you really began by looking at how UV affects yeast, and we were looking at cell survival with UV exposure. So there we were looking at 2 different independent variables, actually. First, we were looking at increased exposure as our independent variable, so increasing amount of time exposed to UV light. For our other independent variable, we were comparing wild-type yeast to RAD1 yeast, which, remember, RAD1 yeast don't have the same repair mechanisms. So with that experiment, we were really trying to address the question of how important are the repair mechanisms in protecting our DNA against damage and cell death. In those experiments, our dependent variable, we're always counting colonies, which we used as a measure of cell survival. For controls, think about positive and negative controls as giving opposite ends of the spectrum. So perhaps a positive control would be yeast that we would expect to have high survival, such as yeast that we did not expose to any amount of UV light. Our negative control, we would want yeast with low survival. So those would be the yeast with our maximal UV exposure. Perhaps even RAD1 yeast with maximal UV exposure would be the most negative control. And then we've, of course, had controlled variables, such as we kept things at the same temperature, we let yeast grow for the same period of time before counting them, etc. For techniques, we used sterile technique. You had to plate microbes, you had to do serial dilution, you exposed to UV light, and counted colonies. And of course, for data analyses, it was really important that we normalized or standardized our data by not just using straight colony counts, but first converting those to percent survival to control for any variation. Perhaps we did a weird serial dilution, so that would help control for that. And then when we looked at our baseline graph from group to group, we were able to combine replicates by taking averages. And when you take averages, you always want to keep into account the standard deviation when you present that data as well. For the yeast, you continued your projects by looking at UV protectants. So every group had a different question, but perhaps you were looking at can coconut oil protect against UV damage? So there your protectant would be your independent variable, same dependent variable as before. And your controls might now look something like the positive with high survival would be yeast not exposed to UV light again, but your negative control might be yeast exposed to the same UV light but without any protectant. We used the same techniques, and the data analysis was again similar. For the choosy worms, everyone again had a different question, but perhaps you were looking at host pathogen interactions, whether C. elegans preferred this bacteria over that, if this particular gene was involved in sensing bacteria, along those lines. So your independent and dependent variables, for dependent variables for many groups, it would be, number of worms on one side of the plate versus the other, so an indication of preference. For some groups, they might have used survival as their dependent variable, so number of surviving versus dead worms. And independent variables were really perhaps different worm strains or different bacterial strains. Controls varied group to group. Perhaps positive control would be a plate where you would expect the worms to show a strong preference, so a plate where you have nothing plated on one side and, say, a nice healthy food source like OP50 on the other. We would expect worms to show a strong preference to go towards the food. Negative control, you might have OP50 plated on both sides. We would expect absolutely no preference between the sides there. For data analyses, we used statistics, so you'll want to brush up on those, whether you prefer the t-tests or the binomial, whichever you prefer is fine. But think about, controlling for plates that got a ton of worms versus plates that got few worms, how you would analyze your data in those circumstances.